It started with mosquitoes. Not the itchy kind you swat on summer nights, but the ones that have killed more humans than all the wars in history. Malaria, yellow fever, dengue. Viruses carried not by bullets or borders, but by wings less than 1/10 of an ounce. In 2015, scientists working out of the University of California created something many believed was impossible. A genetic edit so dominant, so persistent, that it refused to play by the usual rules of inheritance. They called it a gene drive. And the game of life was never the same again. A typical gene, whether for eye color or disease resistance, follows Mendel's laws. Each parent contributes to half. But a gene drive breaks that symmetry. Using a molecular tool called CRISPR-Cas9, it copies itself onto the corresponding chromosome in the offspring, overriding chance. Instead of 50%, it spreads with near total certainty, sometimes reaching 95% or higher of all future generations. Imagine a genetic trait that forces itself into the bloodline again and again and again until the entire wild population carries it. In lab trials, gene drives turned female Anopheles mosquitoes sterile within 11 generations. That's the same insect that transmits malaria to over 200 million people a year, killing more than 400,000, most of them children under age 5. That's the promise. Eliminate a disease vector by collapsing its reproductive chain. But that's only one version of the story. Other teams have aimed gene drives at invasive rodents on island ecosystems, at crop-devouring beetles, even at reversing genetic conditions in mammals. The goals, they vary. Eradicate, suppress, repair. But the method stays the same. Force the trait, break the odds. And this is where science meets the hard wall of ethics. Because gene drives don't stay local. Once released into the wild, there's no border checkpoint. A mosquito doesn't need a passport of any kind. An altered genome can cross continents in months. And once it's out, there's no easy way to get it back. Ecologists have warned of unforeseen ripple effects. Predators losing prey, parasites losing hosts, or wild species gaining traits that become new problems of their own. Resistance mutations where CRISPR site is altered and the drive fails can evolve too, sometimes leading to unpredictable outcomes. Governments and researchers have responded with caution. In 2016, the National Academies of Sciences called for strict containment protocols and public consent before any environmental release. Projects must include reversibility plans, so-called daisy drives that fade out over generations, and ongoing monitoring to track unintended effects. Because even the most targeted gene drive can't predict every consequence. What gene drives offer isn't just power over pests or pathogens, it's power over inheritance itself. That kind of leverage has never existed before, not in agriculture, not in medicine, not even in eugenics. For the first time, we can write traits not just into an individual, but into the wild, into nature's default setting. In 2020, a gene drive test off the coast of Western Australia aimed to eliminate an invasive mouse species, threatening seabird colonies. It worked in simulation. But critics warned that if just one edited mouse escaped the island, native populations on the mainland could collapse in less than 5 years. Because once you change the default, the future becomes a reflection of that decision. It is, by design, irreversible. These are Interesting Things with JC.